Most of it is, it's really, it was like poems. We have to
I
read
might-
poems in Spanish.
I might-
Why do we have to read
How do you-
poems in Spanish?
Okay.
I don't know, I'm hoping
Yeah.
they'll just give us.
No, I
a
was
whole
wondering very
introduction.
yesterday, saying how
There's a bunch of here tomorrow. I'm like,
like she
I
can
don't know.
read, but also like she gets paid and she doesn't. And then she's like, apparently it's not one. I swear he was how she.
All right, guys, let's get started.
No.
So, just a quick little calendar check stuff. Again, we're gonna have another in-class assignment on this coming Wednesday, so make sure you guys come to class on Wednesday. Just so you guys know, we're gonna have 6 of these, right? 2 now, 2 for the next section, and 2 for the final one. For these, I keep the 5 highest scores, I drop 1. So you miss one, it's not too bad. You miss 2 or more, then it's gonna bring your grade down, right? So please try not to miss these tests. So we're gonna be here on Wednesday. If you do get sick or something happens, just let me know. The other one is you guys' schedules. So I scheduled the office hours for this class for Thursday, 11:30 to 1, and then Fridays, 2 to 3:30. If you can't make it to those, these are my office hours for my other class, for development class, but if no one's there, just stop by, then you can come in, ask me questions. And then remember, on top of that is, you can add me on Google Calendar, create an event and you invite me. And then the final way is just stop at my office, right? If my door's open, come on in. With that being said, when I was looking through this stuff, like, when I looked at you guys' names, like I threw everyone's email onto this thing to look for them just kind of like this. A lot of you guys haven't set your, change your privacy settings and stuff. So just so you know, everyone else can see what you put on your calendar. Yeah? So, if you search it, like, if you put one of your friends' name and you can see what they're doing, like, whatever they put in, you'll see their name. To clean it up, do this. Click on these little dots, go to settings, and go down here to this, who you're making it available to, and just leave it on this. Instead of see all event details, just leave it to free or busy, right? And that way, we won't know what you guys are doing. But yeah, because I was trying to look through you guys' email, like, I was trying to pick an extra office hours today, when's everyone free? I caught this thing on there and it populated without, like, it doesn't say beer, like, it was like you guys' stuff, and I was like, this feels weird. So, get rid of it, yeah, you know me, cover it, like, put it into, change the settings, yeah. And let me see. Cool. Let's start. So today we're finally gonna start being able to get into the cool things, or the cool molecules. The first one, of course, are gonna be our nucleic acids. And for this, we're gonna be talking about 2 of them. I'm gonna try to stay on the slides, but to be honest, I'm gonna be making my own little table on the left, and I'm gonna try keeping up with my table, yeah? So for the nucleic acid, basically it's gonna be, the first big one we're gonna be talking about is gonna be our DNA, right? Deoxyribose nucleic acid. This is gonna get turned into, or some of it is gonna turn into this thing called the ribonucleic acid. And you're gonna see this in little cartoons that you're gonna notice is this one's always gonna draw something like this. When it's double stranded, your RNA can only exist in a single strand. The big things about them in general are DNA, this thing is huge, right? Base pair wise, you could be hitting millions of base pairs for your whole genome, and every cell is gonna have a copy of your whole data set. This thing is double stranded. And then the other thing is because it's so big and it's long, for us in eukaryotes, we keep it inside of its own compartment, which is the nucleus, right? So this thing is gonna stay in the nucleus. And that's where it's gonna leave up its life. And this one's gonna set a little bit different from our RNA, right? Our RNA are relatively, they're smaller. They can be a few hundred to a few thousand base pairs. This smallness gives them the ability to go from the nucleus and moves into your cytoplasm, right? Which allows it to do its things like ribosomes and blah, blah, blah. We'll go a little bit more into that, but small, as it get out, it can move. This one, we're gonna find it. Important things about it is it's gonna be single-stranded. This is my little drawing there. To make it more accurate, I should probably make this one. This thing's ginormous, this should be a small little thing. this is the big overall things of how they're working. Functionally, what we're looking at here is this molecule is built with little monomers that you put together to make it a huge long polymer. As we glue the little monomers together, we're going to be bringing in different letters, right? Which are these guys here. Normally, for sure, we always do T, A, C, and G. So with these, you basically can organize them in a specific pattern, and it'll let you create specific tasks. So it's almost like these things are your alphabet. You can put them in a sequential order. It's kind of like we have our alphabet, we put them in a certain order, we can make words out of them. Our cells know how to turn this DNA code into RNA code. It'll turn into a specific amino acid, right? And it'll give us that protein that we really care about. So the big thing up here is we're using these 4 base pairs, but functionally, we're storing data. So that data is going to be like a cookbook, has all the information. It's like our parents normally, when they're making us, one of them gives us 1 chromosome, the other gives us a 2nd chromosome, and we're using their data sets, right? Combining them, and they give you, and depending on what they bring, because let's say you're like, your dad had info from his dad and his mom wanted to make him, he only has the chance to give you half of that data set. So sometimes you get pieces of your grandma, pieces of your grandpa from his side, and you combine them with your mom. And then depending on how those codes are working, you might look like somebody more than the other. Right? But that's all coming from our DNA storing all that data, because that's going to give you the information once what you're supposed to be doing, or what your cells are supposed to be doing. So data storage, here functioning, this one is for gene expression. So if you imagine that the DNA behaves like this really huge, long thing of, it has technically like a full cookbook, like it can make every single recipe you can imagine, right? What we do in our cells is different cells will express different chapters of this piece of DNA to perform a specific task. So like your kidney cells, they'll be putting different proteins out that are helping move ions, right? While your eye cells might be making different pieces of DNA to help it get to a crystal form, so you can see a little clear structure from your eye. So this is, all the cells have all the information. The reason that they're different, that they behave differently, is because at this level, they're going to change the way, which ones are going to be turned on or off. So you might have little libraries. This one has a few genes, this one has a lot of genes. This will be the big function room. All right. Now let's get into why. We're going to nucleic acids, but I'm going to talk about this for 1 second. Don't take notes here. I just left it up here so I remind myself. So I think biology takes a lot of memory. There's a lot of vocabulary, there's a lot of words, a lot of concepts, where when I was studying for these classes and you have limited amount of time, limited amount of energy, limited amount of memory, I normally didn't care about stuff like what year people made it, figured it out. The only thing is either we know it or we don't know it. That's all that matters to me. So for my quizzes, you don't have to remember any names and you don't have to remember any years. I'll never put that on the test. If I do, send me an email afterwards, be like, hey, you said you weren't going to do this, and then everyone gets perfect score on that question because I don't want you guys to study them, right? So you don't need to memorize names. You don't need to memorize years. So we got enough stuff to remember. But we'll appreciate what they did, right? So for this one is when they were trying to figure out these nucleic acids, they found this interesting that they kept getting equal amounts of A and T, and then C and G. And as we go into today's lecture and talk about how the DNA works and how the RNA works, you'll see why they actually picked up those numbers. Right? All right, let's go. So we're going to start off with DNA. So this was their proposed model of how it looked. So back here, they had, you see these little rings? Those are basically our sugars. And then they had little phosphate groups. So like sugar, phosphate, and then you had another thing on the other strand. And then these ATCGs were the little things that were kind of like the little rungs in the ladder. Those are the ones that kind of clamp into themselves. Right now we're going to go a little bit more into all these little shapes. But that's the big goal of what we're trying to get to. To see how all those structures come together and how they work. All right. So again, sorry, this guy is telling me the whole story. right? DNA is in the nucleus, has all your information. Then your cells go through a process. Through transcription. This is this thing right here, right? And what we do from there is we use this double-stranded DNA. We're going to rip it open, and we can use these different bases to match up our bases to get the information out. So from this, we use it as a template, and we'll create a complementary RNA strand. And that's where it's going to take us from double-stranded to a little single-stranded little fragment. Right? From there, this is the things we're going to talk about on Friday. We have translation, which is these codes get read in sets of 3, and the 3 base pairs will dictate what amino acid we're going to put together. And that's how we're going to build a chain of proteins. We get exactly what they're going to look like. Those proteins are going to be the things that are going to be able to do different jobs or tasks inside our cells. So for now, DNA transcription gets us to RNA, and translation gets us to protein. This is central dogma. If you ever take a class with me in the future, going up upper grade, I always bring this up. So this one, memorize it now, and it'll save you a lot of trouble later. DNA transcription, RNA translation, protein. All right. So let's look into the DNA itself, into the single subunit of how it works. So just one single unit is called a nucleotide. This is what we're looking at here. For this thing, you're basically looking at 3 different components that come together. Right? So first, this is somewhat color-coded, right? Here we have our phosphate group, which is that right there, right? P with the double bond of those, and the O's with your negative charge. This is going to be your starting site. Your next big player here is going to be our sugar ring. So we have a pentyl sugar, which has 5 carbons. Whenever you guys count these, I'm going to draw it like this. There's your oxygen. This carbon that's right there, that'll be carbon 1, carbon 2, carbon 3, carbon 4. And you all see that it kind of pops up a little bit. And you see that CH2, that's our carbon 5. So 1, 2, 3, 4, 5. That's our pentyl sugar. Right? For these numbers, you're going to have to learn them. I promise you, going forward, people love to quiz on these things. And as we go to today, I'll have you guys kind of write down what each one of these things are. So we have that sugar kind of at the center. Then we have this, which is our nitrogenous base. So for this is the one that we have different types. And as we go through today, we're going to go through the different types. But the critical thing is, I'm going to have you guys remember how many rings they have. I'm not going to ask you to draw the molecule. Remember that there's like an NH group sticking out on this carbon. You don't have to go to that level. The only thing I want you guys to know about them is a little bit like this. Like, there's your adenine, guanine. You see each of these has 2 circles inside, right? Like carbon circles and nitrogen. These slight differences, like this NH versus this OH. So a double bond O. A double bond O will give us a negative charge. And the N will have a negative charge, but you'll feel positive because you have a bunch of positive hydrogen around. Those slight differences mean that when our when our cells have other molecules trying to interact with this, they'll go and they might be set up to be like, hey, I want to interact with a negative molecule, they can clamp on. But if this is present, because that has a positive charge, it won't be able to clamp on. So these little bonds give us like almost like a little signature of what makes them special. And then all the interactions that comes afterwards are going to be based on how they set up for what letter. So for this is the only time we're going to talk about how the atoms are set up and the polarity. From now on, the only thing I want you guys to remember is you have 5 different bases, right? So in DNA, we use C, cytosine on the left, T on the right, A and G. And these actually pair into each other. And normally, the way that we pair them is going to be A and T make a pair. So when I write my stuff, I always write it like this. A and T will be our first set. C and G are the ones that pair into each other as well. You can see we have one purine, one pyrimidine. That's our next set. They pair up. So if you see ATCG in your sequence, you know you're talking about DNA. When you go to RNA, so let's switch on this. That the A, which is our a purine is gonna wanna interact with a pyrimidine, but instead of using T, it's gonna switch over to U, right? So now our pair is gonna be A, will pair up with U, and of course, C and G basically stay there, right? So that's one little difference between the two. They're gonna swap out this guy. So on the right, it's my little trick of how I remember it, because they'll ask you this sometimes. They'll be like, oh, a purine or a pyrimidine, and you gotta know whether you're talking about a C, a T, an A, a G, or whatever letter they want. So on top, I remember this: to build a pyramid, you have to cut one stone at a time, which I know is kind of long, but in my head, it's to build pyrimidines, right? Which are the ones at the top. It uses 3 different letters: C, U, and T, and that's our cut word, right? So to be cut and in one ring and one stone at a time, it reminds me of that it's only one ring. And by that process of elimination, I know I have the opposite, which is purines, and that's A and D, right? So pyramid cut, and then you'll be on. Does that make sense? Or no? Yeah. All right. So I know they make them look kind of funky in my test when I draw them or I put them on there. I'll make them look super simple like this. Right. So they'd be like, here we have our pyrimidine, and here we have a purine. Pretty cool. All right. Cool. So there we're going to catch our first difference, right? We have the different bases between DNA and RNA. It's going to be one place that we switch. The other one, we're going to go in and we're going to focus on the sugar itself, right? So that's where this chart's going to be a big player. So if we map, little task this thing can do is our first carbon, that's the one that's responsible for holding on to the nitrogen base. Right? So that's one job that we have here. Nitrogen base. This second carbon is going to decide, it's going to tell you whether you're working with DNA or if you're working with RNA. So I could literally draw one of the molecules, right? And what you're going to see is a sugar ring. Remember, normally they have, for every carbon, they'll have an OH group and an H group, right? And here you see it's flipped, but you still have an OH and an H group. So this is a regular sugar, the 5 sugar ring, we call it ribose, and that's basically going to be our RNA, right? So if there's an OH present, we know we're talking about RNA. Deoxyribose just means a ribose without a sugar. And that's exactly what you're missing there. So instead of having an OH, now you just have 2 H's on that carbon. So that's a big difference, right? DNA, you're gonna find an H group. So if I draw a molecule, you guys should easily be able to tell. If you see the OH, it's ribose. If it's the H, then you know it's DNA. That make sense? Yeah? All right. Then the last 2, I kind of want you to remember them as a set. This 5 one is super important with this. So these basically are going to be the places where we're going to anchor and create the chain. So when we're talking about this, we're going to try to tell us what bases and what order they're going to show up. They're often going to write that something like that, where it's going to say 5', right? But what they're referring to 5' is we're starting off at this carbon and we're going that way, right? And there you can see there's our phosphate, there's our sugar, there's our nitrogen, like our first base. Then this will be the one for the following molecule. There's your phosphate at the 5', and then this is your 3', 1, 2, your third carbon, which links up to the following. So then together, your 5' and 3' is how you make the connect your monomers, right? So that's how you're able to make this polymer strand. You're doing covalent bonds at these sites, and you basically plug them into each other. And then the cool thing is, now, depending on what you put on this right side, whatever your nucleoside is, right, for that nitrogenous base, that'll tell us what is written in that DNA code. And because it's in a strand covalently bonded, it's a pretty good thing of being permanent, right? You can't even glue these things together. So for this, before I go ahead, feeling pretty good, everybody? carbon ring, yeah, number in it. The other one is the three groups, phosphate groups, your sugar, and your nitrogenous bases. Careful, because people love using this terminology for these guys. So add these to your little dictionaries, make sure you're following them. That if you're talking about a nucleotide, I'm talking about everything, right? Phosphate, sugar, and this. When we switch over to our nucleoside, we basically ignore this, and we just care about these two, right? And then the last one is we care about the sugar phosphate backbone. We're basically canceling out the nitrogenous base and just talking about this. They love to talk about it in the sense of how is the information actually stored. Right? And the thing is, in the sugar phosphate, the phosphate and the sugar, if it's a DNA molecule, they'll all look exactly like each other. Right? So technically, that doesn't give us any information of like, is this making one gene or a different gene? All the information is actually coming from this side. Then nitrogenous base, which again, is going to change wherever those groups are lined up, will be the code that we have it. Like maybe it's positive charge, negative charge, negative charge, positive charge, or whatever your sequence is. Right? But that would be the thing that we can now recognize and know what we're supposed to be doing. All right, cool. So right now we're walking down one cable on one side, right? Phosphate at the 5 prime, sugar, which is 3 prime, connecting us, and we're walking down the sugar molecule. These now basically represent our different bases that we saw before. And then here we're seeing this pattern show up, right? A and T and C and G. But in this sense, what we're looking at is that interaction is actually happening because you have a second strand, which is anti-parallel. So it's the same thing, but it's kind of flipped on its head. Because now the 5 prime is here, and you have 3 primes. Right? So 5 primes on top, now you have 5 primes at the bottom. And what these do is these match up at the center or like the rungs of the ladder by creating hydrogen bonds. You remember hydrogen bonds, where they're coming from? Those polar bonds, right? And they're spaced out perfectly to the point where A and T are the only ones that want to bind with each other. And these little 2 red lines are super important later on because it means that they're able to create 2 little bonds. Right? So this one, when you talk about hydrogen bonds, this has 2 of them. The C and G, they line up and they actually make 3 bonds of hydrogen. But that'll be the thing where our goal is to keep DNA and keep it safe. And you want this double-stranded cable to be like faithful, like you don't want it to mess up. So the cells will make this, and they match up perfectly because now you have 2 copies of your DNA. If this thing for some reason say got cut and we lost some of the base pairs, our cells actually are able to go back and they'll fill in the gap. And they can reseed it based on that interaction where if they're like they say this and this was missing, the cell will only be able to throw in the G at that spot, and it will only be able to throw a C because that matches up to the G. That makes sense? So for this, the important thing is for DNA, we have a double-stranded helix, which is kind of what I'm drawing there. The DNA is anti-parallel arrangement, which is just that 5 to 3 prime. Remember, we're talking about the sugars here. 5 to 3, 5 to 3, 5 to 3, and then 5. So 3 to 5, 3 to 5, 3 to 5. That makes sense? Yeah. So if I ask you guys to write this, right, you can actually write it like this: 5 prime, C, A, G, T, and it would be almost the same as if I wrote 5 prime, A, and let's see, yeah, A, C, T, G, right? Because when something's reading it, they'll be like, okay, this is the 5 prime side. That'll be my 3 prime side. And you know this is going to match up to something, and this will be a T. And now, because I know they're backwards, when I draw this one, I know I'll put 5 on this side and 3 on this side. So look, T, G, A, C, and look, 5 prime, C, A, G, T. So that nomenclature of putting a 5 prime and then putting your sequence is something you're going to see super often going forward. In most of the classes, they love writing it, so make sure you feel comfortable with it. My eraser, guys. And just in case you guys are wondering, back to this, hydrogen bonds, A and U also make 2, C and G and the RNA also make 3 hydrogen bonds. But the cool thing is, again, like the DNA, when I think about it, is like you're storing your information this way, right? Like CA, CB next to each other is the important thing. There, you glue that through a covalent bond. Like it's not gonna fall the holes, it's not gonna rip off. In this middle section, or like this little connection side here, this is where you have like a duplicate copy, and you're holding that together through a hydrogen bond, which is a weaker interaction. It's easier for you to open it and close it. And then later on, you're gonna see we'll go back into how that helps basically with this process.
Do we always like prioritize reading DNA like 5' first rather than 3'?
So what's gonna happen is, as a writer, you should make sure you always include the orientation, right? So I'll give you the trick. Sometimes I mess with you guys on your midterms. I'll do it on purpose just to mess with you, right? So I could say like 5', and then I'll give you a sequence like this. And then I'll tell you, hey, what sequence would match up or be complementary to that? And what I do is I'll write it like this for you. Right. So the instinctively, when you're looking at it, you're like, oh, this is the exact same sequence. It's not going to work, right? But what you'll see is if you flip this over, these A's will pair up there, and these T will pair up there. But sometimes I mess up, and I'm not a good writer, and I forget that we have to keep writing these things all the time. So some cases you'll get it where people accidentally leave them out like that, right? And there you have to use your best judgment. Normally just kind of, you can say, either you can assume that this is the 5' side, or you'd have to make a comment that, hey, this is ambiguous, it's not clear what this is. Right? But yeah, most of us, we assume with each other that we're all writing 5' to 3', but like if this was on the midterm, you better ask me, be like, hey, dude, you messed up on question 4, like this doesn't have a 5' side, and you'll see I'll fix it right there in the class.
So if it was 3' with the second answer, that would match up.
Say it again.
So it was like 5' AATT. If it was 3' AATT, that would be the correct answer.
Yeah, hold on, 5'. You see, this was my question, right? A, T, T, and I'll put this base pair just to make it it's not a count number anymore, right? 3'. So then here the choices I would give you is like, now we know this is going to match up with your G, and this is your 5' side. So we know it's A, T, T. So this would be the right answer, right? 5' to 3'. But because I'm a buddy and I like to mess with you guys, I'll write your answer like this. Is this being 5' G, A, T, T, right? That would be the right answer. Or I'll write it like this, 5' T, 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 A, G. So on your test, be on your toes, and just know that I'm trying to trick you, right? So here, instinctively, you're thinking, oh my God, this matches up perfectly. A to T, A to T, T to A, and C to G, and you're like, this is the answer, let's go for it. But because the 5' is on the wrong side, basically, you're wrong. Right? So focus on them. My goal is I'm gonna be good about them, and I'm gonna keep them there. So make sure you're vigilant on them, yeah? All right, any other questions? I'm just writing down. All right, so again, cool thing here, our cable, we're connecting it covalently this way, making our long strand to make a polymer that holds our data, is more permanent. This interaction has hydrogen bonds, the little red dashes, which would be that weak thing that we can potentially rip open if you close another one. Here's a fancy version of the DNA. But again, we can appreciate things pretty fast. There's a sugar ring, right? Carbon 1, 2, 3, 4, 5. So you can see this is the 5' end, phosphate. 3' connects to the next monomer, the next monomer, the next monomer. So we build this chain. On this side, look, the phosphate is the one sticking out. So we know this is the 5' end, and we're working our way back. 3, 5, 3, 5, 3. Then the pairing, right? Again, this is the thing that they're really kind of showing off. There's your 2 rings for your pairing, your 1 ring for your pyrimidines, and look, A and T means that there's 2 little functional groups underneath that. There's C and G, those are going to be these three different polar bonds are going to give us that energy to hold this thing together. And it's easier, kind of like you can even just get this DNA hot. You get enough energy, it'll break the hydrogen bonds, and these two cables will come undone. But when they're coiled together, they kind of do this kind of arrangement. They'll twist onto each other, like a spiral. You can compress them, or you can expand them, but they normally like to stay in that spiral sense. All right. Here, I was just going to mess with you guys again about how to label these. So here, I labeled this one like how we uncommonly, like not the way we normally see them, where they always put the 5 prime. You can see here, I was trying to trick you guys. I put the 5 prime down here. Right. So again, the O, and you go around the circle, right? At the first one, first one would be here, is where you expect your nitrogen base, right? Carbon 1 is nitrogen, so now we're looking at it flipped, right? Then you have this second carbon, which I don't know if you have an OH or H group, because that will tell me whether it's DNA or RNA. Our third one is linked up here, and our fifth one is on this side. So now I know that this is the 5 prime, and we're going towards the 3 prime side. So now we see when we read this, we would say 5 prime C A G T to the 3 prime. On this strand, right now it's 5 prime A C T D. That makes sense? Yeah. All right, cool. How are we feeling about DNA and RNA? Making pretty good sense? All right, do me a favor. I want you guys to make this table, but you're going to leave me a little bit of space down here, because this is the stuff we're going to talk about on Friday. We'll show you how we're going to go from this RNA. It's going to turn into protein. So the green stuff is the big info of how this molecule behaves as a whole, or like its properties. The red things are really telling us about the jobs that they're doing, right? Or like the role for us. And then these bonds become super comfortable with them. I want you to know that there is complementary pairs like A and U match up, C and G match up, C, G, A, and T, and these hydrogen bonds. So make that table look nice, and then be ready for me to mess with you guys with the sequences, right? So Kinsley showed me one way I can mess with you guys, right? Which is I can mess with that 5 to 3 prime thing to get in your hands. The other one is I can do something like this. And ask you, is that DNA or is that RNA? I heard you right, RNA, right? The big giveaway is that right here. I had written C, G, P, A, C, then that's the big giveaway, right? That's DNA, and that'll tell us that's RNA. The other way I like to mess with you guys is I might do my own little drawing of the sugar, like this thing, and I'll mess with this second carbon, right? Like I might ask a question like, hey, I'm trying to figure out what this nucleic acid is. It has an OH group at carbon number 2. And you guys have to tell me, so what do we have there? Exactly, orange, right? OH group. What's your name? Elizabeth. I'm trying to put names to read the spaces, right? Yeah. So perfect. Like, just like she said, if I say there's an OH group there, we know we're talking about ribose. If I say there's 2 hydrogens, then we know we're talking about the DNA. Yeah. So what else am I gonna ask you about this? I think that's pretty much it. If you guys don't have any questions, we'll probably call it for today. And then one thing, my goal right now is that after I finish the slide, we'll talk about them. I'm going to write down keywords at the back of the slide at the end of it. And then my goal is when I'm making the quizzes or midterms, I'm going to go back to these keywords. Right? So I'm going to use this as a thing to help me focus on what I should be asking you. So make sure you guys feel comfortable with the words and also kind of how they relate to each other. Yeah. So study the words, give me that table. Another one, if you guys feel up for this, you guys can make me a table for this stuff. I'm talking about if there's a sugar ring, what is each carbon responsible for? Right? So 1, our nitrogen base. 2, it's going to tell us whether we have DNA or RNA. And then 3 and 5 is what's helping us build the chain. Yeah, that'll be good. So that'll be your little at-home self-study assignment. And then again, if you have any questions, stop by office hours or set up an appointment and we can hang out.